Hi, this is Mrs. Bustamante. Today I want to walk us through this animation of DNA replication, the process of DNA replication. So these are all the players that we're going to have to kind of know and know what they do in this process of DNA replication. So this is our DNA molecule. It's a double helix. It would be twisted, but for the sake of understanding what's happening, we typically draw DNA replication as just kind of a straight line. So it's easier to see. We can see that our G's and C's and A's and T's are matched up using our base pair rules, right? And this dark line is going to represent our sugar phosphate bond. So some other things we need to know about. This dark green is our ligase. This light purple is our DNA binding proteins. Helicase, this light green. We have two kind of types of polymerase here. We're really only going to have to know this light blue one. And then we have a primase. So we're going to talk about what these do, and I'm going to step us through this animation to help us understand. So remember, at the start of DNA replication, we have this double helix. What happens next? Well, let's watch. So here we saw, okay, this is our DNA binding protein, and what it does is it basically holds apart our double-stranded DNA because this DNA actually wants to bond together with these hydrogen bonds, but remember, hydrogen bonds are not super strong, so we can actually split them apart, but these binding proteins help keep it apart. And this green right here, this is our helicase, and it's kind of the thing that really cuts open this DNA. I like to think of it as helicase cuts. So as you move down this DNA molecule, it's going to cut it apart. So we take that double-stranded DNA and we turn it into single strands. Okay, then as we move through this step, you'll notice we have this red primase. And up here we can see that on our DNA sequence we had AA, and it added UU. That's one way you can really know that you're adding RNA, right? Because if this were DNA, you would be adding thymine or T's, but instead we're adding uracils. And RNA primase is actually kind of important because we can't actually just add DNA bases to our DNA when we're making a copy of it. We have to add some RNA first in order for DNA polymerase to come along and be able to work. So this is our RNA primase. It's adding some RNA to the strand on the top, and you'll notice it's also adding some to the strand on the bottom. And this is something interesting about how DNA replication works, right? Our helicase is cutting apart our DNA, but you're going to notice that how DNA polymerase moves on the two strands is slightly different. We're going to have a top strand typically. It could be the bottom strand, but we call this our leading strand. And you'll notice that here's our RNA that got laid down by RNA primase. And then right here we have DNA polymerase, this blue square. And it's adding down our DNA complementary base pairs to our original strand of DNA. So sometimes we call this strand that's being made, this new strand, the complementary strand of DNA. So it's adding those complementary bases down, and it's going from right to left. But when we look at the bottom strand, right, we have our RNA, and you're going to notice that goes from left to right. And this strand we call the lagging strand. It does not really move all that much slower than the top strand. In fact, they happen at the same time, and that's important to know. But it gets built in segments or pieces. And you're going to notice a difference when you look at the two of them that way. So here's our DNA polymerase. It's going to add our DNA base pairs, our complementary base pairs. And you can see that this top, our leading strand, looks kind of more complete already than our bottom lagging strand. So here is RNA primase again, and we're going to see DNA polymerase come in and kind of fill that hole, and you'll notice that it has RNA and then DNA and RNA and DNA, whereas our top strand just has some RNA and DNA. So what happens next? Well, we actually have another DNA polymerase. It kind of comes in and it changes that RNA back to DNA so that we now are going to have all DNA nucleotides on both strands, your top strand and your bottom strand. And now we have this square dark green molecule called ligase. And I like to think that ligase is going to come in and it's going to link together my sugar phosphate bonds so that I end up with two solid DNA molecules. So notice those holes will kind of get fixed. And this is my end result. I get two DNA molecules. If you look closely at them, they're identical. And they're identical because we use an original strand that is a complement to the other original strand, and then we basically match our base pairs, right? So this bottom strand here, well, you will notice is exactly the same as the original strand on the bottom, and vice versa. The top strand on the bottom DNA double helix is the same as the original top strand. And that's because we're adding complementary base pairs on complementary strands, meaning they all match up. So these two strands are actually identical. You cannot tell the difference between them. And we say that we will get half from the original DNA and half of this is newly made or synthesized DNA. And so we call this the semi-conservative model.